Tyson from Virginia Tech. Dr. Tyson is a university distinguished professor of biology. He's a mathematical cell biologist, systems biologist, and throughout his career, he's contributed tremendously to our understanding of the cell cycle, as well as the dynamics, timing, and different dynamical properties of various network motifs that we can find in biological systems. Thank you so much for joining us today.
Thank you, Thomas. How do I get out of this so I can get my...
There we go.
Oh, okay.
I'll close this one, and I'll let you find the one.
Go to the desktop.
Desktop, okay.
And here. Okay. Yeah, great. Thanks for sticking with us to the end here. This will flip back and forth for a few more times. It's a great pleasure to be here and to talk to you about the regulation of cell cycle, the cell division cycle in yeast. And over the course of the next 30 minutes, I would like to try to convince you that progression through the cell cycle is governed by a complex signaling network that consists of a number of bistable switches that are embedded in other bistable switches. So that's our goal. This work has been done in collaboration with Bela Novak and Stefan Helt, who are in the Department of Biochemistry at Oxford University. And it was done last year while I was on sabbatical at Oxford, courtesy of Virginia Tech and Merton College. The work has been supported by grants from the BBSRC to Bela and the National Institutes of Health to me and my collaborators at Virginia Tech. So, first of all, the cell division cycle, as most of you know, is the sequence of events by which a growing cell replicates all of its components and divides them more or less evenly between two daughter cells, so that the daughter cells inherit the machinery and the information necessary to repeat the process. The most important component of the cell that needs to be copied and partitioned to the daughter cells, of course, the genome, the DNA molecules. And during the cell cycle, in eukaryotes at least, these DNA molecules are carefully replicated during S phase of the cell cycle. And then later, the products of replication, which are called sister chromatids, are carefully partitioned to the two daughter cells so that each daughter cell gets one and only one copy of each chromosome. So you can conceptually divide the cell cycle into four phases. There's S and M, the copying and collating function, separated by two gaps, G1 and G2. Now, the very first thing I want to claim is that cell cycle progression is not a clock. Under normal laboratory circumstances, it appears to be a clock because the cells are well fed, they're given everything they want, they have all the permissions that they need, and they rapidly grow and divide in a periodic process. But that's not usually the case. There are only very unusual situations in which cells function like an autonomous clock. Rather than a clock, it's much better to think of the cell cycle as a copy machine. It has two functions, as I said, to copy the DNA molecules and then to collate those copies into separate piles, which are going to be the nuclei of the daughter cells. Now, the copy machine in the biology department office, if we supply it with lots of toner and blank paper and then have a steady stream of exams going into the machine, of course it will just function like it's a periodic process, right? making many, many copies of all of these exams. But really the crucial feature of that copy machine is actually what happens when it makes mistakes, okay? There have to be surveillance mechanisms in the copy machine that look out for all the things that can go wrong and stop the copying process. So when you run out of toner, you got to stop and correct the error. When you run out of paper, you got to stop and reload the bins. If there's a dreaded paper jam, you have to stop and give instructions about how to get rid of the paper jam, okay? So these are the really crucial features of a copy machine, and the cell cycle works in a very similar way. There are surveillance mechanisms that keep track of the things that could go wrong. So for instance, if the DNA is damaged during G1 phase by ultraviolet radiation, say, you have to halt progression through the cell cycle and repair the damage before you copy the DNA, because you don't want to copy damaged DNA and pass it on to future generations. And similarly, at the end of the cell cycle, during M phase, the crucial requirement is that all those replicated chromosomes are properly aligned on the metaphase plate. That's called the spindle assembly checkpoint, that the spindle be properly assembled. If it's not properly assembled, you have to block further progression, you have to fix the mistake, and then you can pick up where you left off and continue the process, okay? So what we really want to focus on here are not the periodicity of the cell cycle, but the checkpoints that make sure that cell cycle copying and collating is done properly so that mistakes are not propagated to future generations of cells. Now, conceptually, we'll divide the cell division cycle into two phases, which we'll call interphase and M phase. Interphase consists of G1S and G2. That's the copying phase of the cell cycle machine. M phase, or mitosis, is the collating phase, when all the copies are segregated to the two daughter nuclei. The collating phase is characterized by high levels of a protein kinase called cyclin-dependent kinases. And these protein kinases phosphorylate target proteins in the cell and drive all the events of mitosis. When mitosis is properly completed and you're beginning to segregate the replicated chromosomes to opposite poles of the spindle, then the cyclin subunits of the cyclin-dependent kinases are destroyed, the kinase activity drops, and counteracting protein phosphatase activities rise in the cell as the cell exits mitosis. The job of the counteracting phosphatases is to erase all of the phosphorylations that were carried out by the kinases as you go into mitosis. They all have to be removed when you come out of mitosis so you can get into the next cell cycle. Then, as the cell proceeds through interphase, going into S phase and G2, there's a rise in kinase activity, a fall in the counteracting phosphatase activities, until eventually it's time for the cell to go back into mitosis, okay? So that's the basic underlying structure, and what we would like to understand are some of the molecular details of how all of this happens. Are you with me? I'm not speaking too fast, I hope. Okay. Now, everything I'm not going to show you any equations, and I'm only going to show you one experiment. But you have to take my word for it. Everything I tell you is true, unlike some of the things you hear nowadays. And it's all backed up by detailed mathematical modeling and analysis and simulation and experiments that test the predictions of the model and totally confirm the predictions of the model, okay? But that's all found in our published papers, and you can look it up. What I want to emphasize are the molecular logic of the control system. So it's going to be a lot of molecular biology, but underneath the glitz on the outside is the real machinery of modeling, I assure you. So how are cyclin-dependent kinase activities regulated? Okay, as I said, the job of a cyclin-dependent kinase is to phosphorylate target proteins, and the target proteins then carry out the events of the cell cycle. Okay, so this is the control machinery. The cyclin-dependent kinases, they're like the conductors, okay, and the woodwind section has to do this, and the violins have to do that, okay? And these commands are given by protein phosphorylations. Now, how do we control the activity of the cyclin-dependent kinase? First of all, by controlling the availability of the cyclin subunits. The kinase subunit is present in excess, but the cyclin subunits come and go, as their name suggests. They're cycling proteins. Their levels rise and fall in the cell due to regulated synthesis and also regulated degradation. Furthermore, cells regulate the activity of cyclin-dependent kinases by these inhibitory molecules, which are proteins that can bind to the cyclin CDK dimer and inactivate it. And the active kinase can also be inactivated by phosphorylation of the kinase subunit and then reactivated by dephosphorylation. Okay, so there are many different ways to control the activity of the cyclin-dependent kinases. We also have to keep track of the counteracting protein phosphatases in the cell because they play an equally important role, and I'll talk about their regulation when the time comes. Okay, cellular decision making. I want to talk about signal response curves, and by that I mean suppose in the laboratory we have some sort of a signal, a hormone, a growth factor, or even say an inhibitor of a particular protein in the cell. We treat the cell with these signals and we want to see how the cell responds. In this case, we're thinking about, say, a transcription factor that drives the synthesis of cyclin subunits. That would be the signal, and the response is the activity of the cyclin-dependent kinase in the cell. And in a simple signal response curve, we might see this kind of response that we call ultrasensitivity, meaning when the signal is small, there's no response. As the signal increases, the cell responds actually very rapidly over a narrow region of the signals, and then the response saturates. This is a very typical kind of signal response curve that can be generated by many different types of underlying molecular mechanisms. We call that ultrasensitivity. Its property is that it's a reversible switch. As the signal increases, we turn the switch on. As the signal decreases, the switch turns off at the same place where it turned on. So we'll call that a reversible switch. That's not the kind of switches that we see in cell cycle regulation. Rather, they're irreversible switches that are based upon positive feedback. So here's the simplest example where the cyclin-dependent kinase is inactivated by phosphorylation, and the inactivation and activation are governed by positive feedback loops. And as a result of the positive feedback loops, the signal response curve has this characteristic S shape, which means there's a stable, steady state with low response at low signal levels. There's a different stable, steady state with a high response, and in between, there's an unstable, steady state. And now as we increase the signal, it has to get up to a high enough level to flip the switch on. As the signal decreases, the switch stays in the on position. And if the signal stops there, we say the system is bistable. It has two different stable steady states separated by an unstable steady state. Okay, so it's that paradigm of bistable switches and irreversible transitions that lies at the heart of the regulation of progression through the cell cycle. And that's because the mechanism that controls the activity of cyclin-dependent kinases is full of positive feedback loops. Okay, so if you remember the diagram I showed you earlier, which didn't have all of these dotted lines in it, okay, that was the underlying network of chemical reactions. The dotted lines are all the feedback signals, and most of these feedback signals are positive feedback signals. And each one of them has the capability of creating a bistable switch. We'd like to know which switches are functional and how are they organized to govern the progression through the cell cycle. Okay, you with me? Good, still awake? Okay, now here's the one experiment I wanted to show you. This was the crucial experiment that gave us this idea back in 1991, actually. The paper was published in 1990. It's a signal response curve for the activity of cyclin-dependent kinase as a function of total cyclin level. I'll tell you just quickly how the experiment is done. You make an extract of frog eggs that has all of the proteins in it that regulate progression through the cell cycle, except for cyclin. The cyclin has been destroyed. You add cyclohexamide to this extract so it can't make its own cyclin, and then you add a recombinant cyclin molecules to the extract, which have been clipped off. The part that governs cyclin degradation has been clipped off the cyclin B molecule. This experiment was done by Mark Solomon, by the way, in the late 80s. So you add a fixed amount of cyclin. There's neither cyclin synthesis nor cyclin degradation. So that's the signal. That's what's under experimental control. And the response, you ask, well, is there MPF activity or not? And the basic observation is, if you add just a little bit of cyclin, you see no cyclin-dependent kinase activity. You have to add enough to get past a threshold at about 12 or 15 nanomolar, and then all of a sudden you see plenty of cyclin-dependent kinase activity. And that was called the threshold for activation of cyclin-dependent kinase. Okay, this is a signal response curve, and it should remind you of the irreversible bistable switch that I was telling you about. So in 1993, Bela and I published a little mathematical model based on the underlying molecular mechanism, and we predicted that this system would be governed by exactly a bistable switch, and we made the prediction that there should be two different thresholds. One threshold for flipping the switch on and a different threshold for flipping the switch off. That prediction was tested 10 years later when two papers appeared almost simultaneously in 2003, one from Jill Seibel's lab at Virginia Tech, the other from Jim Furl's lab at Stanford. Both laboratories demonstrated the two different thresholds. And they're separated by about a factor of 3 in cyclin B level. And there were a number of other predictions that were made in our modeling paper in '93 that were also confirmed in these experimental papers. I'll let you go to them to see the details. Okay, you with me? Now, in that model, we assumed that the phosphatase that works against the kinase was constitutive. It wasn't regulated. And there's a problem with that. The bistable switch works because you turn the kinase on and off with a fixed level of phosphatase. So you have to choose the phosphatase level correctly to get the switch to work. But the problem is you get futile cycling as you go around the switch. Okay, so in the low system, there's hardly any kinase activity, so you don't do any phosphorylating, and so you don't waste ATP. But when you flip to the higher position of the switch, now you're rapidly phosphorylating proteins, but you're also rapidly dephosphorylating them. You can only stay in that high state if you're willing to pay the price of a lot of futile cycling. Now, we should have known this. We should have predicted this in 1993, that this isn't the way it's going to work, right? In fact, I knew it. I was teaching this in cell physiology course, that usually when you turn a kinase on, you turn the opposing phosphatase off. That's the way the metabolic pathways work, okay? So if you have a regulated phosphatase and you turn the phosphatase on and off, as you turn the kinase on and off, then you get rid of the futile cycling. And the way you have to do this is you have to downregulate the protein phosphatase by the protein kinase, okay? We could have predicted that. It turns out to be true. It was discovered by the experimentalists just a couple of years ago. The protein phosphatase is a type 2A protein phosphatase with a partner called B55, and it's regulated by a stoichiometric inhibitor called ENSA, and ENSA has to be phosphorylated to kill the phosphatase. ENSA is phosphorylated not by the cyclin-dependent kinase, but by a kinase called Great Wall, but Great Wall is activated by the cyclin-dependent kinase. So it's kind of Rube Goldberg machine. The kinase that we're really interested in activates Great Wall activates ENSA, and ENSA kills the phosphatase, okay? And then, furthermore, there's apparently another feedback where the protein phosphatase inactivates Great Wall. That creates a positive feedback loop that creates bistability on the protein phosphatase, okay? So both the protein kinase and the protein phosphatase are controlled by positive feedback loops that create independent bistable switches, okay? And experimentally, you can knock out one or the other and things work just fine. But when you knock out both of them, then mitosis is really screwed up. And yeah, and that was shown in this experimental paper in Current Biology, published in 2016. Okay. So where am I? 20 minutes. Yeah, I'm doing okay. So we have this complex mechanism. Here's all the molecular details about the positive feedback loop that controls the cyclin B-dependent kinase activity as you go into mitosis, and controls the B55 PP2A phosphatase activity as you come out of mitosis. The two different bistable switches are negatively regulate each other, okay? So the kinase switch turns off the phosphatase switch, but the phosphatase is the counteracting phosphatase over here. It turns off the kinase switch. So this is all very nasty looking, but we don't have to worry now about all the details. Once we understand it, we can kind of reduce it to this simple motif, okay? We have a positive feedback loop on the kinase side that creates a bistable switch. We have a positive feedback loop on the phosphatase side that creates a bistable switch. And those two bistable switches inhibit each other, okay? They're mutually inhibitory, okay? And together, they control the phosphorylation state of all the M-phase substrates. So they control going into mitosis and coming out of mitosis. So now, back to the paradigm of the copying and collating machine. In the collating phase is the high kinase phase when this switch is in the high kinase state, and then as we leave mitosis and go into interphase, the copying phase, that's a high phosphatase state. And Yeah, okay. So next I want to talk about the surveillance mechanism. So what I've showed you so far is we can get the cell to go back and forth between the two phases, the copying phase and the collating phase, by this pair of bistable switches. Now I want to talk about the surveillance mechanisms, and we'll talk about the spindle assembly checkpoint first, okay, which looks for these replicated chromosomes that are not properly attached to the mitotic spindle. How does that surveillance mechanism work? Okay, so in metaphase, when the replicated chromosomes are being captured by the mitotic spindle and aligned on the metaphase plate, there's a complex process where you're trying to catch the replicated chromosomes by microtubules that come from opposite poles of the mitotic spindle. And when they're properly captured, it looks like this, and the microtubules are pulling the chromosomes apart, but the chromosomes are held together by these little rubber bands called cohesin proteins. And so the centromere is under tension, and the cells can tell when the centromeres are under tension. When they're under tension, they inhibit the dissolution of the kinetochore attachments, because if they're properly attached, you don't want to break them. Okay. The problem is when you're improperly attached, when you're attached to one side of the spindle but not to the other side of the spindle. That's trouble, because now if you go into anaphase, you'll pull both chromosomes to one pole, okay? So this is the error that has to be corrected. And the way the error is corrected is when there's no tension but there's attachment, you promote the detachment of the microtubules. And you go back to this state, and then you try again. So it's like a fishing expedition where you're trying to catch, where two people are trying to catch the same fish, okay? So we can think of this also as a bistable switch. There are three states, okay? Neither side attached, both sides attached, or only one side attached. And these two have to be stable steady states, and this one has to be an unstable steady state. So basically, this error correction module at spindle assembly is a bistable switch, and you can see it in the underlying molecular machinery that's trying to correct these errors. Now, as long as you're trying to correct the errors and the centromere is not under tension, it creates a protein complex called the mitotic checkpoint complex, and the mitotic checkpoint complex inhibits another protein complex called the cyclosome. The function of the cyclosome is to destroy cyclin B when you exit mitosis, okay? So you want to inhibit the cyclosome because you're not ready to exit. And the cyclosome also destroys a protein that's inhibiting the protein that clips the rubber bands. So if you think about what's happening here, this is the proper way to do things. As long as there are errors, you're preventing progression from metaphase to anaphase to telophase and out of mitosis. Now, this is also a positive feedback loop. So there's a bistable switch here in the checkpoint pathway, and these two pathways negatively affect each other. Okay, so we have exactly the same sort of motif. We have an error correction module with positive feedback loop creating two different stable steady states. And we have a spindle assembly checkpoint motif with a positive feedback loop that controls whether the cyclosome is active or inactive. And these two positive feedback loops, these two bistable switch inhibit each other, okay? The unattached kinetochores inhibit activation of the cyclosome, and the cyclosome inhibits the bistable switch governing error correction through these feedback loops. So now the situation looks like this. We still have M phase and interphase. Here's the basic motif that's controlling the kinase activity and the phosphase activity. Inside the high kinase state is exactly the same motif, which is governing the surveillance mechanism for the spindle assembly checkpoint. Okay? Yeah, I'm going to be done in time. So we have a pair of bistable switches embedded inside another pair of bistable switches. And I'm almost done because there's actually a third pair of bistable switches embedded in the next phase of the cell cycle. So now we have to come out of mitosis, go into G1, and implement the DNA damage checkpoint in G1. Now, I'll do this a little bit faster. The DNA damage checkpoint is also controlled by two bistable switches. One, oops, I didn't want to go that far. Okay. One of them we call the restriction point, the other we call the G1S transition. At the restriction point, before you go into G1, you have to start making cyclins, the cyclins that govern the G1S transition. They're controlled by a transcription factor called E2F. And E2F is inactivated by a protein called the retinoblastoma protein. And in order to pass the surveillance point, we need some cyclin-dependent kinase activity. It's due to a cyclin called cyclin E. So cyclin E is kind of a starter kinase that can start to inhibit the retinoblastoma protein, remove the inhibition of E2F, and then E2F can create more of itself. That's a positive feedback loop, and create more cyclin E. So there are positive feedback loops on this side that create a bistable switch. Either RB is high and E2F is low, or RB is low and E2F is high. Okay, so that's the bistable switch at the restriction point. Once this switch is flipped to the E2F on state, then E2F drives the synthesis of cyclin A, and cyclin A-dependent kinase is the one you need to do DNA synthesis. But cyclin A-dependent kinase can also phosphorylate and inactivate RB, so we have another positive feedback loop here. And there's a third loop because E2F makes EMI1, which inhibits the cyclosome, which has been killing cyclin A. So again, positive feedback loops creating bistable switches. This has all been proven experimentally by beautiful experimental results published in the papers that are cited here. And again, the motifs are the same, just different players, two sets of positive feedback loops creating two bistable switches. In this case, the interactions are plus-plus interactions instead of minus-minus interactions. DNA damage works upstream by creating a cyclin-dependent kinase inhibitor that inhibits the first bistable switch. We won't go into the details. Here's the final picture. Back to the cell cycle, the high kinase state, the high phosphatase state, governed by this pair of switches, the spindle assembly checkpoint governed by this set of switches, the DNA damage checkpoint controlled by that set of switches. It's switches embedded in switches, QED, thank you for your attention, And the details are in this paper just published in Current Opinions in Systems Biology. Thank you very much for your attention. There's one back here first.
Thank you. I would like to ask about virus infection. They hijack this machinery. Where do you think they go inside? The surveillance or the copying stage?
I'm sorry, I didn't quite.
I'm talking about the viruses when they
Viruses.
Yeah, when they take a cell, they actually hijack the system to replicate. Where do you think they go inside? Where do they hijack?
Which part do they hijack?
Yeah, the surveillance part or the copying part? What do you think?
Well, I think they want to leave both of those parts intact. Where they want to go is convincing the cell to re-enter the cell cycle because they need all that machinery to copy themselves. Okay, so most of our cells are just stuck in G1 because they don't have the right permission slip. So I think what the viruses are doing are giving them permission to replicate the DNA. That's my guess. Well, we don't really study that. I think we better go to another question.
I have two very modest questions. One is, how species specific is this? And the second is, how did it evolve?
Okay. These basic mechanisms, these basic motifs seem to be present in all of the types of eukaryotic cells that we've looked at. The proteins aren't always well conserved. Most of this was worked out in budding yeast, although the actual genes and proteins in budding yeast are not related to phylogenetically to the analogous genes in mammalian cells. But the underlying motifs and networks are the same. And that was what I was emphasizing, I hope, is the molecular logic, not the names of all the players. How did it evolve is a wonderful question, and I don't think anybody knows the answer to that. We would have thought it evolved from prokaryotes, but the prokaryotic cell cycle is controlled by completely different mechanisms that share no analogies what's happening in eukaryotes. The best guess is that in the early eukaryotes, this evolved from, I think, the cytoskeletal machinery of kinases and phosphatases that controlled the phosphorylation of the cytoskeleton. And somehow the circular chromosomes of the bacteria got attached to motor proteins and cytoskeletons, and then you could have more than one chromosome, basically, and still make a sensible partitioning. That's the best story I can tell right now.
Thank you, great talk. I've been out of the literature for a while now, but if I recall correctly, positive feedback loops are associated with the augmentation of genetic noise and production of. So one of my questions is, do you think, because would you expect then, because there's so many positive feedback loops within this control mechanism, that genetic noise would be a huge issue, and cells have to be really, really good at doing something about this? Thank you.
Yes, that's a great question, and I didn't address it at all. I've only been talking about deterministic models. But we've been worried about this for the last 10 years, and we spent a lot of our efforts converting our deterministic models into stochastic models and seeing how sensitive the system is to noise, because we worried about exactly that same thing. The problem, as you know, is to get a good stochastic model, you need good data on single cells, which wasn't available until recently. It's the subject of a different talk, how these networks behave in a stochastic setting. And you'll just have to take my word for it that they seem to be robust enough. It's pretty nice they are no more or no less robust than real yeast cells, which is a good place to be. You don't want them to be too noisy, but you don't want them to be too reliable either, because we have single cell data that tell us exactly how much noise can be tolerated, right? The system can be noisy because the surveillance mechanisms protect you, right? But you have to make sure the surveillance mechanisms aren't too noisy, so that you can correct the errors, and when the errors are corrected, then you can pick up where you left off and continue through the cell cycle. But it all seems to fit together and make a very convincing story. I think everybody's exhausted. I am. Thanks for a great day. Thomas, you were going to say some final words about the whole...
So, on behalf of SysMod organizers, I want to thank everybody for attending today. Please subscribe to our Google Group mailing list. We're going to try to, as years go on, we're going to try to make it more active, more interactive. We also had a really good discussion over lunch about what we can do to provide to the community in terms of resources in computational biology. So, as organizers, we will try to facilitate that, we will try to do that, but also please become active members of SysMod and help us make the whole community better. Before you go, don't go